In codominance, we actually get to see both traits, kind of a mix of it, but not like the paint. Where in a paint you would see like red and white make pink. In codominance, you would see speckles of red and speckles of white. So in here, our first example is using chickens. Blue is codominant with white. Blue chickens are BB, white chickens are WW, and chickens with the genotype BW show a checkered pattern, where you get to see both the blue and the white colors coming through. So cross a blue with a white. So with our offspring here, our blue offspring again would be BB, and our white offspring, sorry, our white parents would be WW. And so in our Punnett square, when we do that one, we would get R's on the brain there. BW. Now, could you write it WB? Sure, why not? We usually just do it the alphabetical because that's our habits of the English language. So our genotype ratio, the only genotype that we have here is BW, and that is a 1:0. Our phenotype, the only thing that we would see is the checkered pattern, and again, that would be in a 1:0 ratio. So cross two chickens from the offspring in problem 10. And so our only offspring here are our checkered pattern, and so we would write out our Punnett like this, and it's very similar to what we did with the incomplete dominance, except you just have to keep in mind that when you look at these, you would see both colors. And so our B's are, sorry, I did it again. Yeah, so we'd have BB to BW to WW, and really you could do these in any order. This is just kind of an old habit of mine. Our ratio there is 1, and I misdid that one, to 2 to 1. And our phenotypes are blue, I'm just going to put check, to white, and so that would be, again, 1 to 2 to 1 ratio on that for our codominance. And so this one's pretty easy to work through. Now when you get to the bottom here, we get to blood type. Now blood type's kind of interesting because on our little blood cells, if you know somebody who is a type O, they just have a nice little blood cell with nothing really on it. Our I's, though, up here, IAs and O's, the I stands for immunoglobulin, because on some little blood cells we have little immunoglobulins, little proteins with the letters A on them. So this would be type O, this guy would be type A, and then we also have some immunoglobulins with the letter B, so this would be type B. And then the fourth type has both immunoglobulins with A's and B's on them. So when we look at people's blood types, if we mix them together, we have these little antigens that go and attack things that don't belong in the body. And so if you're a type A person and we put type B blood into you, your antibodies go and attack that. And that's not good because it causes coagulation, which is clumping, which can cause blood clots and do lots and lots of damage. If you are a type B person and you get type A, you're in trouble as well. But if you're a type AB person, you can actually have receive type B blood because your body doesn't attack B's because you already have it. You can receive type A blood, you won't attack that one either. And you can also receive type O because it has no letters on it and the body doesn't really even recognize it to attack it. Type O people are up a creek because if you put any of these other blood types in there, it's going to do lots of damage. The antibodies are going to go after it and clot up again. So there's some kind of cool information on that, and I'll post some links on the site to help you understand that concept a little bit better. But when we write these, we do it with the letter I and then a superscript. And so you'll see I's with the A's, meaning they're holding type A's. And so a type A person would have AA, or they could inherit A's and O's. Type B person would inherit both B's or a B and an O. An AB would inherit the immunoglobulin A and immunoglobulin B, and a type O person would only inherit the little immunoglobulins with the O's, which means they have nothing on them. And so if we were to cross a homozygous type A, so that means they are IA with a type O person, we always assume the type O is homozygous because that's the only way we can receive it. So IO, it's off to work we go. So IA, IO, and when you work these out, we usually do the dominant one, or it's kind of a co-dominant, incomplete dominance. It's a crazy thing with blood types, but we would do the A first and then the O. This person would show as type A blood. This person, or potential offspring, I guess you could say, would also be a type A, as would this one, and we would also see this one. So in this case, our genotypes, the only genotype we can have is the type A, the IA with the O, which means that if you're the offspring and mom's type A and dad's type O, in this case, you are definitely a carrier of both the A and the O blood types. So this is a 1-to-0 ratio, and type A, again, would be 1-to-0. Let's look at the next page here, question 14, where we're crossing two ABs. The only way you can be AB is if you are IA and IB like this. So IA, IB, I don't know why I keep doing that, IA, IB. Potential offspring here, this would be a type A. This one would be an AB. Here you would have, that would be an AB person, and this would be a type B person. So our genotype ratios, IA, to IA, IB, and IB. In this case, we have 1 with A, then the AB is 2, and then the BB is 1. Look at that 1-to-2 ratio. It comes up a lot in this. And then our phenotype ratio of A to AB to B is also 1-to-2-to-1. Now where it gets scandalous is, let's say you had two parents who were AB and they have a child who is type O. There's something weird going on there. So mom and dad might have to have some discussions, or I think somebody got switched at birth. And so these are going to work out pretty much the same. And actually here down in 17 is kind of a fun little who gets the inheritance and who gets to be thrown in jail. And you can work through these. So I've got the key at the front. Come on up and see me when you're done.